Good morning. Today I'm going to present on functional anatomy of skeletal muscle. So why I choose this topic is because by definition of exercise is a contraction of skeletal muscle that produce the bodily movement. So the most basic thing that we need to know is the skeletal muscle. So muscle itself in our body, there's three types. Cardiac, skeletal and smooth. So the skeletal one is the voluntary one, and the cardiac and the smooth is involuntary type lah. So mainly the smooth muscle cells usually found in the digestive tract or blood vessels. Cardiac muscle cells usually in the heart, and the skeletal one usually in the muscle, bone, and tendon lah. So the next thing I'm going to proceed is I'm going to talk on the structure of the muscle lah. So it's very important for the contraction of the skeletal muscle. So the basic structure of muscle is basically skeletal muscle. It's covered with outer connective tissue, which we call it as epimysium. Okay, so this is epimysium. So this epimysium is an outer connective tissue. It has a dense, irregular, fibrous connective tissue. So this one is further compartmented into muscle vesicles lah. So this muscle vesicles outer part is perimysium. So it also has this connective tissue. Okay, see this one also has a connective tissue. It's a dense one, irregular and fibrous lah. So it's the same as the epimysium lah. So and it's further divided into it further compartmented to muscle fiber lah. So this muscle fiber is also known as muscle cell lah. So we have this covering muscle fiber. We have something covering outer part is called it as sarcolemma lah. So it's in Greek actually. We call it muscle, sarco, and lemma is a sheep actually. So it's in a Greek word actually call it. So this is basic structure of muscle, which is important in the contraction of the skeletal muscle lah. So the next I'm going to go further in, which is the muscle fiber. So which was called as a muscle cell. So if you see, there's a lot of nucleus here, right? So usually what is different from the other cells is multinucleated. So and also it has if you see there's around stripes here. So this muscle cells usually has a striated appearance. So which the striated appearance, why we have the striated appearance? Because of the A and the I bands lah. So that one I will talk later on in the contraction of skeletal muscle. So this is the why we get this striated appearance lah. So further in is a myofibril with the sarcolemma outer part lah. So actually there's a endomysium endomysium, and covering the endomysium is the sarcolemma lah. So the endomysium is also a connective tissue. It's an areolar connective tissue. So difference between the epi, peri, and endo. The epi and peri has rich in collagen, but the endo is a bit less collagen lah. So what that makes difference lah. So why we need all this? Just because of the production of the movement actually lah. So there's further in, I'll go further detail. So which is if you see, this is a myofibril lah. So further in there's a myofibril here, okay, which has the thick filament, the red color, the blue color with the thick filament lah. So these are the other structures we have here. So this is the other myofibrils, and all this is we call this the yellow color. If you see here, this is all the T tubules. So it covers all this myofibril. So it's like netting out, like it's covering the whole myofibrils. That is T tubule, and sarcoplasm is similar to cytoplasm in a normal cell, but in a muscle cell we call it a sarcoplasm lah. So this is the muscle membrane and the terminal system. Okay, so there is actually triad this is triad, actually call it triad. So what is this triad? Triad comprises of the sarcoplasmic reticulum and also the T tubules, this one. So this thing and this thing, so this combination of this thing is called as triad lah. So what does this sarcoplasmic reticulum do? It's similar to the similar to the smooth endoplasmic reticulum lah. So in muscle cell, it stores all the calcium in lah. So that is main function of the sarcoplasmic reticulum lah. So there's a T tubules here, if you see. So there will be a calcium regulation later. I'll be talking on. So yeah, this is just a rough picture first. Okay, so like what I said, plasma lemma and plasma lemma. So it's a plasma membrane lah. So this is what we call a sarcolemma, is a plasma lemma with basement membrane. So it has a junctional fold. So this junctional fold is the impulse transmission from a motor neuron to the muscle fiber. So other function is to maintain the acid base balance and transport of metabolites from capillary blood into the muscle fiber. So it has this satellite cells lah. So the satellite cell involves the function as a growth and development of skeletal muscle adaptation to the injury and immobilization and the training lah. So sarcoplasm is like similar kind of cytoplasm. It's a gelatin-like substance fills the space between and within the myofibrils. So all the contents is dissolved proteins, minerals, glycogen, fats, and necessary organs. So what differs from the cytoplasm is it has a large quantity of stored glycogen, and also it has oxygen binding compound myoglobin. So it works something like a hemoglobin lah. So the next one is the transverse tubule. Like I was telling you, there's an extension of the plasma lemma that passes literally to the muscle fibers. The picture I showed you just now. So the function is like sending impulse received from plasma lemma to the myofibrils, and it provides pathways from outside to inside, enabling the substance to enter the cell and waste products to leave the fibers. So sarcoplasmic reticulum is very important because it's a solid site of calcium lah. So mainly for the muscle contraction. So the next thing I'm going to talk is myofibrils. So myofibrils, the one muscle fiber, like what I showed you the picture just now, has about 10 to thousands of myofibrils. So and it further divided into the sarcomere. So sarcomere is the basic contractile element of the skeletal muscle. So the next thing I'm going to talk is about the sarcomere lah. So okay, this is a bit complicated. Okay, this is the thin filament and this is the thick filaments. Okay, so the thin filaments, there is actin, troponin, and tropomyosin. Okay, so if you see the thick filaments, it has the myosin. So first I'll focus more on the thin filaments lah. So they have this thin actin. So actin is this. This is the F-actin. This is the actin monomers. So how this actin is being produced? So from the G-actin. G-actin is also called as actin monomers. Monomers will polymerize to F-actin. So means this is one molecule will polymerize to F-actin. Means you'll have two other molecules like that. So this is what we call as F-actin. So the G-actin is called as globular actin. The F-actin is called as filamentous actin. Why is it called filamentous? Because it's being chained. Okay, so this is the active site of the actin. Okay, and this is the supporting character. Okay, we call it as nebulin now. So this nebulin works like it blocks the active site first to like from the myosin to come and combine, bond with the actin. So this nebulin will block the thing first. So okay, so this is the nebulin, it's like a supporting character there. So then there is troponin, so and tropomycin. So if what makes this this is tropomycin. So if you see the tropomycin is this like a trait kind of thing. This is tropomycin. Okay, this is tropomycin. Okay, and this also as well, so it will avoids the our active site from the active actin who bind from mycin, the tropomycin as well. And nebulin, like a stabilizing character, like a stabilizing filament lah, protein lah. So troponin has three things lah. So this is the troponin. Okay, troponin has three complexes. So we call I, E, and C. So the I is usually bind to the actin, and the T usually bind to the tropomycin, and the C usually bind to the calcium. Okay, so this is the what was I was talking about, the steam filament lah. So then you can see a T. Okay, this is the bigger version of it lah. So if you see from here, there's the M line. So M line is accessory protein, we call it. And between this end of this, if you see this is a H zone actually, like we call it H zone. And there's a A band and I band lah. So sorry, we'll go for. Okay, so this is A band lah, we call it. So and this one we called it as I band lah. So the end of myosin filament with another end is called it as A band, and the one with the Z1 and the end of the myosin filament, we call it I band lah. So why is it called as A band and I band is because of any anisotropic, and also I because of the isotropic. So I means two light bands lah. So A is anisotropic, means dark bands lah. So okay, that's like basically about actin lah. So then there's other supporting characters, which is the titin actually. So titin will connect the end of the myosin filament to the Z line. So that is titin. And then this is the M line, this is the H zone. So this from one Z zone, the Z line, to another Z line, we call it as a sarcomere. So that is sarcomere. The whole thing here is we call it as myofibril. So that is about this whole thing here lah. So then we go to this thick filament. So there is thick filament here. Okay, you see this is the one actually, the black color thing. So they zoomed it, and then okay. So this thick filament, we usually call it as a myosin molecule. Okay, I label it as a head and arm and as a tail. Some people call it as S1 or S1 to S2 junction or S2 tail. But for me, I just prefer to say as 8 arm tail. So there is six polymers actually combined to this thing. So which is the 2 light chains and 4 light chains and 2 heavy chains lah. So the thing is that what makes there is in this S1 globular head, there's ATP actually lah. Over here it says ATP actually lah. So this tail here, there's like a helical strand actually like a helical strand, this tail. So all the light chains is here actually, the heavy chains is here. So this one is S1 to S2 junction. We call it as a hinge, or sometimes we call it as arm lah. So okay, this is the titin. Okay, so basically that is about sarcomere lah. Okay, so this is like one thick filament. Myosin is one filament formed by 200 molecules lah. So each myosin molecule consists of 2 protein strands, which I already explained just now, twisted together. One of each strand is folded into globular head. So the myosin head will be the one forming the cross bridges during the muscle contraction. So this is the titin actually. So it stabilizes the myosin filament along the longitudinal axis. So it extends from Z disc to the M line. And this is about the thin filament I was mentioning just now, actin, tropomyosin and troponin. So nebulin is like a supporting, like anchoring protein, so mainly for the mediating the actin and myosin interactions. So then this is about tropomyosin, which already I mentioned, and troponin lah. Okay, so the next that I'm going to touch is about the muscle fiber contraction, how from nerve goes to the muscle. So the main thing here is the alpha motor neuron lah. So it's a nerve cell that connects with and innervates the muscle fibers. So this alpha motor neuron. So previously I mentioned in previous presentation on motor unit. So a single alpha motor neuron will send impulse to muscle fibers. This all is roughly about 300 to thousands of muscle fibers lah. So then what we mentioned is about this neuromuscular junction. So from a nerve, an impulse being sent to the muscle. So there is a synapse or gap between the alpha motor neuron and a muscle fiber. So okay, so there's a dendrites, there's an impulse being sent. So the dendrites will collect the signal, and the axon will pass the signal through the myelin sheath, and then it will pass to the muscle fibers. So this is where the nerve connecting to the muscle, and there's a junction here, right? So it's what we call as neuromuscular junction. So I will talk about this excitation, contraction, coupling. So this is just a rough picture I'll give. So further before I'll go further in lah. So when it begins with the motor nerve excitation, it results in contraction of muscle fibers. So excitation, contraction, coupling. So action potential from brain and spinal cord to the alpha motor neuron and arrives at dendrites and travels to the axon to axon terminal, and then action potential will arise to the axon terminal, and the nerve endings will release a neurotransmitter which call it as acetylcholine, and this acetylcholine will crosses through the synaptic plaque with bind to the receptors on the plasma lemma. So after enough of the ACH binds to the ACH receptor, so the action potential will be transmitted full length of the muscle fibers as the ion gets open in the muscle membrane and allows the sodium to enter. So this is called as depolarization. So I assume you guys, okay, I'll go further detail a bit more. Sorry. So, okay. Okay. So, this is how the detailed part lah actually. So, from action potential, okay, so the first part here you see from the impulse getting from the spinal cord. So, from spinal cord, action potential being sent. So okay, sorry, I apologize for my bad drawing here, but okay, so the action potential will be traveling around, okay, so to the axon terminal. So this is axon terminal, called as axon terminal. Okay, so once this axon terminal being like the action potential goes in, so there's a channel here. We call as this, you saw there's a square thing here, voltage gated sodium channel. So this channel, there's a 2 here actually. So what will happen is that the sodium will go in, okay. So when sodium goes in, there will be more electropositive inside the cells actually. So then what will happen is that okay, then as if you see there's vesicles here, right? So all this having acetylcholine lah. So okay, all this having acetylcholine. So how this acetylcholine being produced? First thing, so is because of this mitochondria. From the mitochondria, it produces this acetyl-CoA. So this acetyl-CoA and this choline, how this choline comes from? This choline is from this actually from diet lah. So they say it is from the digestion part actually. So this choline will combine with this acetyl-CoA and produce the acetylcholine. So and then it goes to the vesicle. So it with the help of enzyme of choline acetyltransferase. Okay, so then the acetylcholine will go in. So when the acetylcholine goes in, okay, so there's a lot of electropositive charges here, right? So there's a lot of electropositive charges. So there's the red color here marked, right? So this red regulatory protein will make sure the calcium channel also open. So the calcium will go in as well. So then there will be more positive charges lah. So if you see from the graph here, actually this is the action potential graph. So basically this is the time and this is the voltage lah. So in a resting membrane, potential usually will be in negative 90. So means the cells is in a resting period, so negative 90 lah. So and then when there's more positive electropositive here in the cells, so it reaches the action potential lah. So it gets the action potential, so up to 30 millivolt actually. So then this is how those vesicles, okay, so this is what we call as a synaptic vesicles lah. So it's acetylcholine inside, and by the process of exocytosis, the acetylcholine will go out. So and then there is actually how this how this acetylcholine from this thing to this thing, how this happens actually is by this process lah. We have this synaptic vesicles and the acetylcholines here. So there's proteins here actually bind through. So this is what we call as synaptotectin and synaptobrevin. And this one is this is actually is a SNAS lah. We call it as T SNAS and V SNAS. So this is V SNAS and this is a T SNAS. So what will happen is that it will intertwine this protein and this protein will intertwine with this protein, and it will cause the acetylcholine to go out. So that's what we call as so acetylcholine will go through out this lah. So okay, the gap between this one and this one is a synaptic cleft. We call it as a synaptic cleft lah. So what will happen? Acetylcholine will go out. Okay, so then there's a ligand gated sodium channel. Ligand gated sodium channel. So okay, this is we call it as a nicotinic receptors lah. So okay, so the acetylcholine will go and then will bind to here. Okay, so once it bind, this thing here, right, the gate here, right, will be open. So no more gates ready. So it will be open. So more sodium will go in. So when more sodium goes in, more positive charges in. Okay, and also the potassium will go out also lah. So to balance, right. So the potassium will also go out, but lesser. More potassium will go out. So both go in. So that's what makes to the action potential lah, reaching to 30. So then after that, what will happen is that a lot of positive charges here, right? So a lot of positive. So then like I was mentioning about this triad, so the sarcoplasmic reticulum with the tubules. So the one protein that holds this calcium is calciquestrin, it seems. So calciquestrin is the one holding is calcium lah. So calcium this sarcoplasmic reticulum with this tubule lah. So if you see there's how is the calcium is being released here. So it's this receptors actually lah. We call it as a dihydropyridine receptor with this ryanodine receptor. So which is this dihydropyridine receptor will bind with this ryanodine receptor. So will bring out this calcium out. So when the calcium is being out, so then we will go to this myofibrils lah. So that's how this action potential happens. The contraction happens. Sorry. So the next will go for the okay, so that's what is this about, this excitation, contraction, coupling lah. So I already explained like a detailed one just now. So this is the acetylcholine. Okay, this is the small picture of it lah. So acetylcholine released from the axon terminal binds to the receptors on the sarcolemma. So then the action potential is generated and travels down to the T tubule, and the calcium will be released from the sarcoplasmic reticulum in response to the change of the voltage. So then the calcium will bind to the troponin, and cross bridges will form between the actin and the myosin. So this is the contraction. So what happens if there's a relaxation? So then the acetylcholine esterase will remove this acetylcholine from the synaptic cleft. So then the calcium will be transported back to the sarcoplasmic reticulum. Then what happens is that the tropomyosin will bind to the active site on the actin, causing the cross bridge to detach. So this is about the sliding filament theory lah. So if you see, the length will be the same, but the tension will be a bit different. So sarcomere and the sliding filament model of the contraction. During contraction, the myosin will ratchet along the actin myofilaments, compressing the I and H bands. So the A band will be untouched. The touched one will be the I and H band. If you see the difference between the I and the H band, then during the stretching, the tension will be released, and the I and the I bands, H and I bands will expand, and then the A bands remains the constant throughout the length of the myosin. Length of myosin myofilaments does not change. So this is a molecular mechanism of the muscle contraction lah. So there's a four steps actually here lah. So the first is so there's a ATP, like I was mentioning. The myosin head has a ATP inside. So as a new ATP attaches to the myosin head, so the link between this actin and myosin will be weak. So and then the cross bridge will be detached. So means it won't detach lah. Then after that, what will happen is that there will be a hydrolysis, ATP hydrolysis. We will release this ADP and phosphate. So as the ATP is split into ADP and phosphate, the myosin head is energized. So means it has energy. So then it will be cocked into a high energy conformation. So it prepares itself for contraction. So then after that, this myosin head attaches to this actin filament and then forming a cross bridge. So this is what we call as cross bridge lah. So then this one, then it goes down, and then the inorganic phosphate generated in the previous contraction cycle will be released, initiating the power stroke. So there will be a power stroke here. Myosin head pivots and bends it as it pulls on the actin filaments and sliding it toward the M line. Then the ADP is released lah. Okay, so that's all.